The overall goal of this procedure is to dissect the human pancreas in a manner that maintains the anatomical orientation. This is accomplished by removing the duodenum and spleen from the pancreas, then clearing away excess fat and vessels from the pancreas. The second step is to paint the anterior surface with blue ink and posterior surface with black ink. Next, the pancreas is divided into 3 regions. Each region is then sliced crosswise into even slabs and placed onto paraffin cassettes, OCT molds, or minced further and placed into vials. Ultimately, samples are collected from all 3 regions of the pancreas, while the correct anatomical orientation is maintained for future analysis.
This method can help address key questions in type 1 diabetes research, such as the mechanisms underlying loss of beta cell mass or function and the nature of immune cell attack.
Visual demonstration of the pancreas processing method is critical, since maintaining the in vivo orientation of the pancreas requires attention to detail.
Demonstrating the procedure will be Lynda Schneider, histotechnologist, and Emily Montgomery, biological scientist in my laboratory.
Begin by preparing all of the materials needed for the procedure. First, label the cassettes that will hold the paraffin blocks with the donor identifier, sample type, aliquot number, and any additional information. Next, label the OCT molds in the same manner. Cut enough pieces of aluminum foil to wrap each OCT block and set them aside for use later. Prepare all of the vials needed to store the fresh and frozen samples. Add RNA later and RPMI media.